co-president with David Long, who is out of state right now, of the Marin chapter of CNPS. And I'm very happy to have this evening as our speaker, one of this year's Grants and Scholarships awardees by the chapter, Nicole Ibañez. And the topic is climate as a driver of divergence in soil specialist plants. And this meeting will be recorded, so you can revisit it later if there are details that you want to learn more about or missed, and or if you want to encourage other people to see it who might have missed the live program. And we will be opening it up at the end for a live question and answer period. But in the meantime, you could put your questions in the chat. Use the chat feature to record your questions as you come up with them during the program. So Nicole has been studying different phenology or bloom times of the wiry snapdragon, Antirrhinum vexillocalyculatum. Sorry, that's a mouthful. In populations growing on closely on adjoining or nearby serpentine soils and non-serpentine soil habitats in areas of lower and higher precipitation. Knowing the plant's phenological and associated genetic differences can help us to understand how rare fragmented plant communities might be affected by stressors such as more frequent and severe droughts caused by climate change. Nicole is a nature enthusiast, born and raised in the Central Valley of California. She completed her Bachelor of Sciences in Field and Wildlife Biology at Cal Poly, San Luis Obispo in 2016. Here she developed a passion for conservation ecology and a strong interest in California native plants. After college, she moved to Sacramento and began a career in environmental consulting, deepening her knowledge of native plants and wildlife, and providing guidance on natural resource and special status species avoidance and mitigation. Close study of the ecology of rare plants sparked an interest in species that grow on specialized habitats such as serpentine. In 2019, she moved to San Francisco to begin a master's degree program in biology at the University of San Francisco. With the help of her advisors, Dr. John R. Paul and Dr. Sevan Suni, whose labs study botanical evolution and plant pollinator relationships respectively, Nicole has conducted a novel research project on the effects of climate on local adaptation to serpentine soils. Please welcome Nicole. So Nicole, can you share your screen?
Yes, let me get all set up here. All right, can you see my screen? Does that look good?
Yes, looks
okay.
Good.
All right, yeah, thank you for the introduction. My name is Nicole, and I am excited that you're all here today and interested in my research. I will be talking about my project, Climate as a Driver of Divergence in Soil Specialist Plants. All right, I want to start by acknowledging that this research takes place on the traditional territory of the following indigenous tribes: Ohlone, Muwekma, Ramaytush, Coast Miwok, Tamiya Nation, Choqueño, and Concow. More information can be found on this website, nativeland.ca, which is a great website. And it also just happens to be Indigenous Peoples Day. So happy Indigenous Peoples Day. Perfect opportunity to acknowledge the land that we live and work on. So I'll start by talking a little bit about climate change. We should all know by now that the global climate is changing rapidly, and temperatures are rising and precipitation events are becoming more frequent and extreme. And a changing environment means that species need to adapt to new conditions or disperse to suitable habitat elsewhere, otherwise they'll perish. So it makes sense that recent work has focused on investigating species adaptations to harsh climatic conditions. And especially high priority should be placed on species of rare fragmented ecosystems, which are the most vulnerable to decreases in biodiversity due to climate change. So special edaphic floras, or plants that grow on specific substrates, are examples of naturally fragmented taxa that may be particularly vulnerable to climate change. On the one hand, many of these species have special adaptations that help them survive with harsh stressors of their native environment, and so these traits might translate to dealing with unfavorable climates. But on the other hand, these kinds of species may have far lower chances of successfully dispersing to new suitable habitat than like habitat generalists. And so therefore they may be more at risk of extinction. In fact, about 600 of the about 1,700 rare plants in California are associated with either serpentine, limestone, volcanic outcrops, vernal pools, or other special substrates. And that's about 35%, which is pretty high considering the small amount of area that these substrates actually occupy in the state. So for this reason, there is an urgent need to study serpentine flora and other special edaphic flora in the light of climate change. And so that's why I chose this study system. So a little introduction to serpentine. Serpentine soils are derived from serpentinite rock, which is actually our California state rock. This is an ultramafic rock that it gets uplifted from below the Earth's crust by tectonic plate movements. So we often see it on the surface along mountain ranges and fault lines. And these soils are distributed globally, but hotspots occur in Cuba, New Caledonia, and California. So this soil is overall pretty toxic for plant life. It has really high heavy metals, high magnesium to calcium ratios. It's very low in nutrients, and it doesn't retain water very well. So as such, most plants really can't survive here, and these habitats can look pretty barren, as you can see in the foreground of this photo compared to the more vegetated background. And then the sparse vegetative cover further results in habitats prone to erosion and higher soil temperatures, making them even more stressful. However, some plants have special adaptations that allow them to survive on serpentine, where if they can make it, they're greeted with much less competition from other plants. Serpentine outcrops are home to numerous substrate endemic species. And in fact, in California, nearly 10% of all California endemic species are restricted to serpentine, while these soils make up less than 1% of the land cover in the state. So these habitats provide a lot of California's biodiversity. Some species that can grow on serpentine, but can also be found elsewhere on other soils, are called serpentine tolerators. True serpentine endemics are almost always found growing on serpentine. And so that might seem a little strange, because if a plant can survive on serpentine, can it survive anywhere? But it's because some of these adaptations that allow plants to survive the harshness of serpentine have trade-offs with competitive ability. So the traits that help them survive in this environment make them worse competitors when they're surrounded by lots of other plants. And that's all right, because there really isn't much competition on serpentine because they are so barren, but it means that they usually don't grow anywhere else. So that's how they get restricted. Some examples of these traits are slow growth rate, small leaf area, and accelerated phenology. And this just means the timing of flower production. So many serpentine plants produce flowers early in their life cycle. Now, how does climate really fit into all of this? Well, some researchers before me have found a pattern between precipitation and serpentine endemism. They found that serpentine endemics tend to occupy regions with higher rainfall than non-endemic sister taxa. Serpentine endemism is especially high in tropical and Mediterranean biomes, so places where it rains a lot. And even within California, there are more endemics in the rainy north than in the more arid south. So this reported pattern of climate, stress adaptation, and endemism suggests that climate may play a role in driving local adaptation to soils. And I'll explain that a little bit better later, but I want to point out this map really quick. If you aren't familiar with California, then please notice that it is usually a lot wetter up here in the Northwest. And as you travel kind of south and east down the state, we have some really dry regions down here. So we do have this kind of natural gradient from wet to dry as you move across the state. And then this black and white map shows the distribution of serpentine soils across the state. So you can see these little black patches. They tend to follow the mountain ranges. So the Sierra Nevadas are right here, the Klamath ranges, and then all up and down the coast ranges. So we do have this distribution of serpentine soils across most of the state. So this pattern I just described between serpentine endemism being linked with areas of higher precipitation is thought to be tied to this trade-off between drought stress and competition stress. Serpentine species are well adapted to handle drought, and there are studies that show this, but they are poorer competitors where competition is really high. So the theory that has been proposed is that on more fertile non-serpentine soils, competition will be higher where it rains more, but drought stress and competition remain pretty stable across serpentine soils. And the higher competition stress on non-serpentine soil in rainy areas leads to serpentine plants being more restricted to serpentine in those rainy areas. And a really great test of this theory is a study done by Fernandez-Going and Associates in 2013. This figure comes from that paper that they published. And they measured specific leaf area of plant communities on serpentine and non-serpentine soils across this natural rainfall gradient, where they defined small specific leaf area is thought to be more stress tolerant, and large specific leaf area is a more competitive trait. Excuse me. So they found that in dry, stress tolerant trait of small specific leaf area was found on both serpentine and non-serpentine soils. And in contrast, in wetter areas, the serpentine communities still had more of the stress tolerant small leaves, but the non-serpentine communities mainly had fast growing larger leaf plants. So this is an example of a trait being more different across soil types due to the precipitation in that region. I found this really interesting, and I thought that this same pattern could apply to other traits that are related to this drought stress competition trade-off. So the trait I decided to focus on is phenology. This word again refers to the timing of flower production. Phenology has been shown to be linked with water availability and can actually shift in response to drought as an avoidance strategy. And this is a trait that's also been found to differ between serpentine and non-serpentine communities, with serpentine plants usually flowering earlier. And the rationale goes like this. In a dry environment, it's advantageous for a plant to produce flowers early and reproduce quickly before they perish. In these dry environments, there's less competition and therefore less pressure to grow big and tall, but more pressure to keep a small stature, narrower leaves, and reproduce quickly. Alternatively, in a productive habitat with high levels of competition, it's advantageous to devote more resources to growing tall vegetation and wide leaves earlier in the growing season and produce flowers later in the season when water is still available and the entire plant is larger. In productive environments, there's less pressure to reproduce quickly and more pressure to increase vegetation size. And this trait is also really interesting because the timing of flower production has crucial implications for gene flow between populations. So if you imagine two nearby populations of a plant, they may have some gene flow because pollinators can go back and forth, visiting both, sharing pollen, and in turn sharing alleles between the populations. But instead, if two nearby populations flower at different times, we would expect them to have less gene flow than populations that have open flowers simultaneously. And if that difference is large enough, then this could eventually lead to reproductive isolation. So this pattern has led me to wonder if precipitation might be driving divergence between serpentine and non-serpentine populations to become more specialized. And I mentioned why I think flowering time is an important trait because it's influenced by precipitation and soil type, and it has implications for gene flow and therefore reproductive isolation. So I've narrowed this down into two main questions. Is there more divergence in phenology between serpentine and non-serpentine populations in wetter regions than in drier regions? And is there more genetic differentiation between serpentine and non-serpentine populations in wetter regions? So I created this graphic to kind of visualize my predictions. I expect that in a dry region, both serpentine and non-serpentine habitats are going to experience pretty high drought stress and relatively low competition stress. Versus in a very wet region, serpentine habitats are still going to have that higher drought stress and low competition stress, but the non-serpentine habitat in this region would have much lower drought stress, higher competition stress. Because these environmental factors have differences in divergence, more divergent, less divergent, I expect that phenology, a trait influenced by drought stress, would also be more divergent in wetter regions. And because phenology can affect gene flow, I also expect allele frequency to be more divergent. In other words, higher genetic differentiation in these wetter regions. So to answer these questions, I decided to focus on the species called Antirrhinum vexillocalyculatum, which is a mouthful. So we can just call it wiry snapdragon. This is a native species of snapdragon that has a pretty wide distribution across California, mostly Northern California, as you can see on this map. It is a serpentine tolerator, so it grows on both serpentine and non-serpentine soils. In fact, about 55 to 64% of the occurrences are on serpentine soils, so it's pretty balanced. So this spread across different precipitation regimes and different soils is going to be important for my study, which I'll get into next. And it's also an annual plant, which makes it a good candidate for greenhouse study. So I want to remind you again that California has this patchy distribution of serpentine soils shown in black that span almost the whole state. And it also has this natural precipitation gradient shown in the different colors. And this distribution of different soils across different precipitation regimes is the perfect stage for a really interesting comparison utilizing this intersection of soil and precipitation. So I want to illustrate for you my conceptual model for this comparison. I am interested in looking at pairs of populations where one population is growing on serpentine soil and it's paired with a nearby population on a non-serpentine soil. And then these two populations within the pair should be relatively nearby and experience about the same amount of precipitation as each other. Then I want to select multiple pairs like this that come from regions with different precipitation levels, as shown in these 5 boxes. Using the setup, I will be measuring divergence in traits between paired populations and then comparing that divergence across pairs from different climatic regions. And specifically, these traits, again, that I'm measuring are divergence in flowering time and genetic differentiation. So to put this to the test, my study has a few different pieces to it. I'm going to explain each one in more detail. But basically, I got started with selecting sites that fit this conceptual model. And then I got out and performed fieldwork at these sites. Later, I ran a greenhouse common garden experiment to test the divergence in phenology in these populations that is passed on to their offspring. And the last step was extracting and sequencing DNA to test differences in genetic differentiation. So step 1 was finding the right sites. To do this, I cross-referenced soil data, precipitation data, and records of actual plant occurrences to find populations that fit my conceptual plan. And I also had to go out and ground truth these, make sure that populations were actually where they were supposed to be. And I ended up finding 5 population pairs across Northern California to study. So there's 3 pairs here in Marin County, 1 pair down here near San Jose, and then 1 pair up near Paradise, California. So this is the wettest of the bunch. This is the driest population, and then these 3 are kind of in the middle. Next, I headed out to find these populations. And when I did, at each site, I took measurements of soil moisture and vegetation density. And these were really just to confirm my assumptions about the differences in soil moisture and competition between the habitat types. And I also recorded some different plant traits in the field, like the phenological state of the plant when I got out there, how many flowers versus fruits versus buds it was producing. And I also recorded height of each plant, but most importantly, this is when I collected seed samples and leaf tissue from these plants. And I collected samples from at least 30 plants per population. Next, I conducted a greenhouse common garden experiment. So I propagated those seeds that I collected from the field sites and grew them up in the greenhouse under identical conditions. So this is the common garden part of that. I used seeds from 11 different populations. So this was 5 of those pairs, and then I have 1 extra that I just decided to grow anyways. And I planted seeds from 30 different maternal families per population. I was able to germinate about 1,600 plants that went absolutely wild in the greenhouse. The picture on the top left is right after I had germinated them. So this photo only shows half of the plants that I had in the greenhouse. And then the bottom right is the plants all grown up. It's hard to tell, but they're going absolutely nuts. Some of the plants were over 1 meter high, which is crazy. They do not get that tall in the wild. So I had a lot of plants and a lot of flowers to work with here. And so once they were growing, I went in twice a week to take detailed notes on germination, growth rate, and most importantly, I counted the number of buds and the number of flowers on each plant. So this was just to give me a timeline, basically, of when each plant began and ended flowering and how many flowers were open at any given time. And this then allows me to compare the flowering time between these populations and compare the divergence between pairs to reveal any differences in phenology that are being passed on to offspring. Because again, these plants are all being grown in the same conditions. The only difference between them is where they came from, where that maternal plant came from, and the conditions that that maternal plant was growing in. And then to answer the second question of my study, I had to do some DNA sequencing. So I extracted DNA from leaf tissue from the samples that I collected in the field. And I'm right now in the process of running double digest restriction site associated DNA sequencing, or DDRAD. And this will allow me to generate single nucleotide polymorphisms. And this basically just allows me to compare differences in the genetics of these populations. And eventually I'll be able to calculate FST, which is a metric of genetic differentiation. So going back to my main question, my prediction is that if phenology is significantly different in some of these pairs, then I would expect that gene flow is lower and we might see more genetic differentiation between these populations. So doing this analysis would tell me if that's true or not. So I am still working on analyzing all of this amazing data that I've collected. There is a lot of it, so I'm still working through that. I do have some preliminary results figures that I can share, but I want to caveat that I haven't calculated significance values yet, so this data is still preliminary. So what the first figures here are showing, this is data from my greenhouse study. So the first figure is showing the number of days to first flower. So how long it took for that first flower to open on each plant. And then that is plotted against the average spring precipitation of the location where the parent plant came from. So each vertical line is one population, and each point is a plant in that population. The second figure, I've calculated the average number of days to first flower for each population. So I've grouped all the individuals, calculated an average for that population, and plotted that against the average precipitation. And the bars there show standard error. So my takeaway from these graphs is that the number of days to first flower does appear to correlate with the average spring precipitation. The plants from really dry locations appear to be adapted to flower earlier, and the plants from wetter locations are flowering much later. They're taking a much longer time to produce that first flower. And again, this has nothing to do with the amount of water they received in the greenhouse. It was all the same, but this is determined by the genetic component. And you can see in this figure that although precipitation does seem to correlate, there doesn't appear to be a super clear pattern between serpentine and non-serpentine populations. And so in these two figures, I have calculated what I'm calling flowering curves. So this is the total proportion of the population that is in flower, so has at least one flower open over time. So in the first graph, I've plotted the serpentine and non-serpentine populations that are paired in the same graph. Figure 3 shows a pair from a very dry region, and figure 4 shows the pair from the wettest region. And so I do see the pattern that I predicted, where the dry populations have pretty similar flowering curves, and the pair from the wetter region are a little more divergent, with the serpentine population flowering earlier. You can see that that blue curve is shifted to the left a little bit. So that is what I expected to see, but the difference doesn't appear to be that dramatic. And I haven't quite yet tested if this is significant or not. So we will see about that. But regardless, I am thinking that the effect of precipitation appears to be stronger than that of soil type on shifting that flowering time. This really only, again, shows the genetic effect on phenology. Any environmental effects in the field are not being reflected here. So this is pretty cool. If there is a significant difference, that would be evidence of local adaptation. So I hate to leave you on a cliffhanger, but this work is still ongoing. So please stay tuned for the final results. Whether or not I find the pattern that I'm predicting, this research is going to add something to our understanding of local adaptation, habitat specialization and endemism, and as well as population dynamics under climate change. And this is really important because although my study species is common, habitats with a lot of specialization like serpentine are home to a disproportionately high number of rare and endangered species. So understanding how these communities might be affected by differences in precipitation is crucial to predicting how they'll be affected by climate change. And this can inform our conservation priorities for these sensitive species and communities. As you probably noticed, I don't have results yet for the molecular component of this project. I'm still working through that. So that will be really interesting to see how the second question in my study gets answered. And in addition to this research, I'm coordinating with my advisors about some future directions that this research could take. This summer, Dr. Suni and I led a field crew to take field measurements of flowering time as well as pollinator observations. And so this data hasn't been processed yet, but I am excited to see what patterns arise with that environmental factor in play. In addition, I know the SUNY lab has begun a crossing experiment to really dive into understanding reproductive isolation in this system. And so this is really just the tip of the iceberg. There are so many questions that we could ask and so much to learn using the system. I'd like to take a moment to acknowledge and thank everyone who's supported and assisted with this research. Biggest thanks to my advisors and my committee members, Dr. Paul, Dr. Suni, and Dr. Randall. In addition, this work would not be possible without funding from USF, from the Marin chapter of CNPS, that's you guys, so thank you, and from the Whitehead Fellowship. Permit coordination came from Marin Municipal Water District in East Bay Regional Parks. I'd also, again, just like to acknowledge that my field and lab research took place on native lands of several indigenous tribes listed here. I'd like to give a huge thanks to the biology department staff for all their help with everything in the greenhouse and lab, and all the members of the Pollen SUNY labs for their help and support, and especially the people listed here for their hands-on help in the lab, field, and greenhouse. Alec, Kate, Raven, Adelaide, Allie, Brianna, Benny, Victoria, and Tatiana. And that concludes my presentation. So now I would love to welcome any questions.
Okay, it doesn't look like anyone submitted anything into the chat. Do we have any people can unmute and ask live questions if they'd like to?
Well, you ask her. You have to type it, don't you?
No, it sounds like Doreen. I have a question.
Sure.
If you
I don't want
take,
people to see me. I'm here with Miss Spotted.
Ed,
Oi.
can you
I
mute,
don't mind.
and then I'm happy to let you go because you'll have a smarter question to ask. If you take seed from the dry areas, like all the way down in the south, and you plant them in the ground in the wet areas, do they still flower early?
Yeah, that is a great question. And that would be a really cool experiment, doing like a reciprocal transplant, seeing like how that happens. But I think so, you know, what I've done is taken seeds like from these different precipitation regimes, and in the greenhouse, I'm giving them the exact same amount. And so what I'm looking at is just that genetic component of, you know, if local adaptation is occurring, then we would expect that would be something they would pass on to their offspring as well. So even without any change in, you know, the offspring's precipitation essentially, or irrigation, that we would still see that difference in flower timing.
Any idea how long it would last? I mean, that could be quite a interesting way to try to move populations into more adapted spaces and things like that. If you had that genetic, you know, if the pollination was mistimed with the pollinators, or the blooming was mistimed with the pollinators, then if you use seed from a dry area, it would bloom earlier. You would have a longer bloom time. I'm just musing out loud here.
Yeah, like using like what we find in this to kind of manage those populations.
Yeah, to help combat climate change and keep the populations from disappearing.
Yeah, absolutely.
Thank you.
Okay, Doreen, if you'd like to unmute and ask your question.
Did she consider the Mount Verdel population of the Antirrhinum?
Yeah, actually, Mount Verdel was one of the places that I collected, and so that is one of my populations.
So where was the non-serpentine one that's close then? I'm
sorry?
Where was the non-serpentine population that's supposedly close?
Yeah, the serpentine.
I mean, I know where the serpentine one is. I don't know where the non-serpentine is.
Yeah, the non-serpentine one is in an area called Verissimo Hills. I think it was like 1 or 2 miles away.
Okay, so I haven't been to the Verissimo Hills, so I just wanted to know where it was. There's lots of places I haven't been. And also there's a patch on the Fairfax-Bolinas Road near the Water District. Did you see that one?
Yes, yeah, that was another one.
And that's a non-serpentine one. Right?
Yeah, correct.
Yeah.
That's the non-serpentine.
But I don't know where the serpentine one is that's close, you see.
Yeah, the serpentine one out there is along Toy on Fire Road. I think that's the one.
So did Andrea tell you where they were? You know, Andrea from the Water District? No.
Andrea Williams?
Because she knew the plants locations very well. Yes.
That probably would have been a great resource. Yeah, I found.
I mean, which year was this? Because of course Andrea has moved on since, and I'm not very current. You know, I'm not in good health right now, and not very young either.
Doreen, just so you know, Doreen is our former rare plant chair for the chapter. Long time volunteer.
Yes.
So
great.
she pretty much knew where everything non-native and native grew in the
Well,
county.
there's big
and
holes in the
advisor.
knowledge.
Well,
Yes.
really not that many.
That's amazing. Yeah, that would have been great information to know. Yeah, the whole trying to locate these populations was definitely an endeavor. I found a lot of, records and database searches to find them, and then it was a lot of ground truthing, checking them out. I remember there was one population that I got out there, and it was just a field of goats, and they had completely eaten everything. So it was
Yes.
an adventure trying to find
Goats as a method of clearing brush also clear native plants of note. Just
Yeah.
like the highway people, if they go down a road, they'll cut all the natives and leave the French brew. It's prettier or something. Yes. Okay, well, good luck with your research.
Yeah. So the other locations in Marin were,
I don't know many locations in Marin.
yeah. But
Yeah.
Nicole, where were the other places in Marin?
All right, so there was one along Concrete Pump Road, which is like a trail within Marin Municipal Water District. There was that Fairfax Bolinas Road. There's Toy on Fire Road. There were two in, oh gosh, what was it called? Another regional park. I'm blanking on the name right now. I've like abbreviated all the names, and so I don't see it all the time. Yeah, those were the Marin ones.
Neat.
To say not many locations, really.
Yeah.
Okay, we have one question from the chat. Did germination success vary between seeds collected from serpentine soils versus non-serpentine soils?
That is a good question. I haven't compared those numbers and ran the statistics on it, but I didn't see much of a difference in germination just between soil type. No, certain populations did better and others didn't, but it didn't really correlate with soil type, I don't think.
Thank you. And Terry, you have a question?
Yes. So Nicole, great presentation and cool project. I'm excited that you're doing that. My question is a little bit more on your literature search and kind of what you're doing to find out more about serpentine. And it actually, part of that discussion you guys just had regarding goats, I'm interested in land management on serpentine, and there are some specialists that have been restoring serpentine habitats that are promoting goats on serpentine and also promoting actually nitrogen deposition on serpentine. And I just was wondering if you had read any literature that could give us more information about different management techniques. And I guess as a side on your project, when you were collecting your seeds from the wet soils versus the dry soils, did you tend to find any additional organic matter in the wet soils?
That is a good question. You know, I'm not sure about organic matter differences. I mean, I did measure soil moisture, and that was really all I was looking at with the specific soil itself. The rest of the soil information comes from those digital soil data. So that's what's telling me like the type of soil that I'm on. I think it would be super interesting to pair this with an actual like chemical analysis of the soil and really see like what's going on there. But for this purpose, I was more just focused on the serpentine versus the non-serpentine. But yeah, that would be really interesting to find out as well. And yeah, as far as management on serpentine, I mean, most of the resources that I've found aren't specific in like management applications, but a lot of them deal with like, there's a lot of research out there about climate and serpentine, how regardless of this comparison that I'm doing, looking at how different climates and precipitation regimes affect how plants grow on serpentine. And so, yeah, I'm happy to find some of those resources and share them if that is helpful to you.
Thank you. That would be great.
Okay, any further questions? Well, thank you very much, Nicole. This was an interesting presentation, and I'm hoping that you'll come back to us maybe with a little article for one of our e-bullets or e-newsletters when you have the final results, and or have any other information that you uncovered that you'd like to share with us.
Sure, I would love that. And I just want to say thank you again so much for this chapter support. It was really instrumental in me carrying out this project, and I really appreciate it.
Great.
And for giving me this platform to share my research.
Thank you.
You're welcome.
So next month,